matrix, and there are cells scattered in the matrix. And that's very different from epithelial tissues, which are tightly packed cells with very little matrix between them, because the purpose of epithelial tissues is to form barriers primarily and to secrete and absorb. Here, the non-living matrix is the plasma. Now, what's in the plasma? Lots of stuff, right? There's water, there's waste products, there's hormones, there's proteins, there's electrolytes, there's dissolved gases like oxygen and CO2, okay? And all of that provides viscosity and function to the plasma, okay? The hormones are chemical signals that coordinate responses to stimuli that the nervous system doesn't want to have to deal with. The immune components defend against disease. And of course, the transport of nutrients and waste products is critical so that the body doesn't run out of building blocks, a source of energy, and also doesn't build up toxic components to the point where you kill the cells and tissues, okay? So the formed elements are the cells, right? Those are the erythrocytes and the leukocytes. The prefix erythro means red, and cyte means cell, right? So red blood cell, white blood cell, okay? The platelets are called thrombocytes, and their primary job is blood clotting. The platelets and the erythrocytes are anucleate, no nuclei, no DNA in these guys, okay? As a result, the platelets and the erythrocytes don't last very long in circulation. The platelets last a few days, the erythrocytes about 120, and then they have to be broken down and recycled, and then new blood gets generated by the bones of the axial skeleton, the pelvic and pectoral girdle, and the proximal heads of the femur and the humerus, okay? Spun blood is going to yield you 3 layers. If you take whole blood and put it in a tube and spin it down in a centrifuge, the dense stuff goes at the bottom, and then lesser density stuff is going to be on top, right? The plasma on top is going to be about 55% of the whole content. The erythrocytes are on the bottom. They're about 45%, and the buffy coat is about 1%. The hematocrit is the percent of the whole blood that is made up of formed elements, okay? So it's a measure of the oxygen carrying capacity of the blood. The higher your hematocrit, the higher your oxygen carrying capacity, the higher the blood's viscosity, okay? Blood is an opaque fluid. It has a metallic taste because it has electrolytes in it, particularly iron in the hemoglobin, right? The color is going to change depending on the oxygen content. The color in presence of oxygen is bright red, and in low oxygen, it's dark red, okay? And you might say, well, I've never seen dark red blood. Well, that's because the minute the blood's out in the air, it hits oxygen and it turns bright red, okay? But the blood in your systemic veins, which are the blood vessels that bring blood back from the body to the heart, are dark red, okay? And they look blue through the walls of the vein, right? The pH is between 7.3 and 7.45, and it's about 8% of our body weight. If we get above 7.45, we experience alkalosis. Below 7.35, we have acidosis. Average volume, 5 to 6 liters for males, 4 to 5 liters for females. That reflects 2 things: the difference in size, all right, and also the difference in muscle content, okay? Guys tend to have less fat and more muscle because of the higher levels of testosterone. Females, less testosterone, less muscle, and more fat per unit volume, okay? The functions of blood: distributing stuff, regulating blood levels of that material, and protection, okay? So distributing what? Hormones, immune components, nutrients, gases, removing waste products like carbon dioxide and ammonia and urea, okay? Regulating levels of substances critical to survival. regulating, for instance, blood sugar levels, regulating the levels of waste products and nutrients, regulating even its own blood pressure, okay, and blood oxygen levels. And then the protection is coming from the leukocytes, but also from plasma proteins that are manufactured in the liver, such as complement, right? So we have a lot of defense mechanisms in the body, and some of them are what we call our innate immunity, which means that the chemical defense is the same regardless of the identity of the pathogen. And then some of these are adaptive immunity, which is going to be generated in response to a particular antigen and will only attack it. Okay? So it delivers oxygen and nutrients to body cells, transports metabolic waste to lungs and kidneys to be eliminated, moves the hormones around. All right? Maintains body temperature by distributing heat. How does it do that? Well, when we're hot, we see what we call peripheral vasodilation, meaning the blood vessels near the body's surface open up, and we lose more body heat through radiation, and that also causes more sweat production. And evaporative cooling then cools the skin, which cools the blood, which cools the body. All right. It also maintains the normal pH using the bicarbonate buffer system and the protein buffer system. Okay. So that's critical because if the pH goes wacky, right, then we create disease. And if it goes way out of whack, then we die. And the reason for that is that if we have a pH that's way beyond our normal range, we're going to screw up membrane potential, and we're going to denature protein. And those two things are going to kill cells and tissues. Okay, so we only survive within a certain pH window, a certain body temp window, and so on. It also maintains fluid volume in the circulatory system. We have to have a certain amount of fluid in the body for the body to function normally. And part of that has to do with electrolyte balance as well, right? Electrolytes are charged particles in water that can move and conduct current, and also can move and draw water to them when they move. Okay, so the body, in large part, determines where our water goes by directing where our electrolytes go. So the water basically chases primarily sodium in the body. All right. Okay, good. It prevents blood loss through a process called hemostasis, right? You cut yourself, you bleed. If that doesn't stop, you die, right? So what happens here is that plasma proteins called clotting factors and platelets will promote clot formation and will generate a temporary seal over the opening, and then tissue repair can take place under the seal, and then we can finish the job. It also prevents infection. Again, we talked about complement, antibodies, white blood cells, okay? All of these defend against disease, all right? And the body, in areas where it's injured or areas where it's infected, will produce chemical signals that will tell these components when and where to get off the blood supply and into the tissue where it can do its work, all right? Inflammation, for instance, is an example of that. Blood plasma is 90% water. Other stuff that's in it, we've already discussed, right? That's why plasma has a straw color to it after you spin down whole blood, okay? Plasma proteins are the most abundant solutes. They stay in the blood and they're not taken up by the cells. The proteins in the plasma are mostly made by the liver. 60% of this is albumin, whose primary job is to allow the blood to maintain its fluid volume through osmosis. And then globulins are 36%, and then fibrinogen, which is part of the clotting cascade, is about 4%. Okay? So liver whips these out. They're always in the blood, ready to go. That's why people with liver damage have trouble with things like blood pressure, blood clotting, and immune defense. Okay? Because a compromised liver doesn't do those things as well, doesn't make those proteins as effectively. Okay? So that's the composition of plasma. I don't need to go through every component, but just read this over and jot down the important points. Right? This is 17.1 in the older edition of the book. Albumin is going to allow blood to hold on to its fluid volume, but it's also part of the protein buffer system, which is important in maintaining the pH in the blood between 7.3 and 7.45, right? If the liver goes down, then what happens is that the blood will lose fluid volume to the surrounding tissue, and you'll get a phenomenon called edema. Okay, this is particularly pronounced in the abdominal pelvic region, where we see a swelling of the abdomen because of fluid leaking out through the peritoneum into the abdominal pelvic cavity. And so you get this distended belly as a result, and that tells you that the liver is failing, or at least is not up to the task. Okay. Albumin also can carry substances in the blood as well. Okay. White blood cells are complete cells, while the erythrocytes and the thrombocytes don't have organelles in them, right? Most of the formed elements live in the blood for only a few days. The exception to that are certain classes of leukocytes, okay? Blood cells originate in bone marrow, and depending on where they're due to go next, all right, they may or may not divide, okay? We're going to find out, for instance, that the B lymphocytes begin in the bone marrow and then go straight into the blood, and then from there they go to lymphatic organs like the spleen. Some go to the liver. They go to the lymph nodes, okay, tonsils. They kind of hang out in those regions and do their work, okay? The T lymphocytes start in the bone marrow, move to the thymus, and then they get educated on what antigens to attack and what antigens to leave alone. And then those that survive that process are released into the bloodstream, and they go from there to immune organs to do their work, okay? Erythrocytes are made in the red marrow, and they go directly into the bloodstream without stopping off at any other organ first. And then platelets come from cells in the red marrow called megakaryocytes that have an unusual property. They undergo mitosis but not cytokinesis, so the nucleus is very large. And so what they do is they have these cytoplasmic extensions that extend through clefts in the bone, and then little bits of those extensions shear off into the bloodstream, and those are the platelets. Okay, so a platelet is really a cell fragment and not a cell, so to speak. Okay, now this is a look at an erythrocyte, a red blood cell. Looks like a frisbee. Okay, it's got a thick rim and a narrow middle, and the reason for that shape is to increase surface area, because one of the big important processes that helps an erythrocyte function is diffusion. Okay, so the diameters of these red blood cells are such that they're almost the width of a single capillary, which is why when red blood cells go through a capillary bed, they acquire a lot of damage. And so after about 120 days, that damage is enough that the red blood cell has to be destroyed and recycled. Okay. Hemoglobin and carbonic anhydrase are the main proteins inside the red blood cell, and also a protein that stabilizes the membrane. It's called spectrin. Okay. And it contributes to blood viscosity, right? The more formed elements you have in the blood, the thicker your blood is. Viscosity is just resistance to flow, okay? So what are some things that increase your hematocrit and thus your blood viscosity? Well, testosterone, okay? So that's why men tend to have a higher crit than women. Low oxygen levels can produce a condition called secondary polycythemia, in which the red blood cells go into overdrive, or the red marrow goes into overdrive and makes lots of red blood cells in order to increase the oxygen carrying capacity of the blood. Okay, but there's also disease states that can cause this, such as cancer of the marrow. Okay, The biconcave shape, we talked about the fact that that's to increase the surface area. 97% of the RBC is hemoglobin. There are no mitochondria, and ATP production is anaerobic, meaning not oxygen consuming, so it's low, okay? And that allows the oxygen they transport to get to where it's going, right? And that's also the reason why they can't repair any damage. They don't have any architecture to do that, right? They're just sacks, and they move, and eventually the sacks get worn out, and then they have to be broken down and replaced, okay? Red blood cells are dedicated primarily to gas transport. They move CO2 and oxygen and a few protons around, okay? They have a buffering function as well. The hemoglobin is a buffer for the protons in the blood. And their main job is to transport oxygen bound to the iron in the heme, which is bound to the hemoglobin protein, okay? And hemoglobin is an unusual molecule in that it's designed to suck up oxygen in conditions that prevail at the lungs and release oxygen in conditions that prevail at the tissues. So it's a perfect design for that job, okay? Normal values between 13 to 18 grams per 100 mil in males and between 12 and 16 in females, right? And that's, again, reflection of testosterone levels and relative muscle mass. Globin in hemoglobin is made up of 4 polypeptide chains, 2 alphas and 2 betas, and then it has a prosthetic group called a heme, which is manufactured from an amino acid called histidine, okay? This gives blood its red color. The iron atom binds the oxygen, and each hemoglobin molecule can transport 4 oxygens, and each red blood cell has about 250 million hemoglobin molecules. So if you do the math, right, 250 times 4, right, and that's getting you to 1,000 million, right, which is quite a lot of oxygen inside a red blood cell. Now, why? Why do we need this special protein to do this? Well, the answer is that oxygen is a nonpolar molecule and doesn't dissolve very well in water. And so if we didn't have a special sponge to soak it up, we wouldn't be able to carry it anywhere, okay? It only dissolves about 9% in water, and that's not enough to allow our tissues to function. So we need this special protein to do this work, okay? The bright red color is the result of 2 different effects, right? The fact that you have conjugated double bonds in the organic portion, right? Double bond, single bond, double bond, all around, right? Those molecules tend to be brightly colored in solution. And also the fact that you have metal ions in there causes bright colors to take place, right? So you got 2 factors in here that promote the red color of blood, okay? In the lungs, we produce something called oxyhemoglobin, all right, where the oxygen is going to pass through what we call the respiratory membrane of the alveoli of the lungs, which are they're like little air sacs, okay, which the respiratory membrane is made up of the endothelial cells of the alveoli, the basal lamina, and then the endothelial cells of the capillaries. And it's less than a micron thick, okay? So it's very, very thin, promotes the movement of the nonpolar oxygen straight through into the blood in the lungs because the oxygen is flowing down its concentration gradient, that's diffusion. And then it's going to diffuse into the red blood cell where it's going to attach to the iron in the heme of the hemoglobin. And that produces oxyhemoglobin. The unloading in tissues makes deoxyhemoglobin, where the concentration gradient for oxygen shifts and the oxygen detaches from the hemoglobin and moves into the tissue, which is exactly what we want, okay? So essentially, you're looking at a transport mechanism, right? The conditions that exist in the lungs are low CO2, high oxygen, and lower temperatures. And that all promotes oxyhemoglobin formation. The conditions in the tissues are higher temperatures, lower pH, higher CO2 content, okay, and that promotes oxygen unloading by hemoglobin, all right? How does that work? Well, basically, the protein changes its shape so that when it wants to make deoxyhemoglobin, it's in more open shape, and the oxygen is more likely to get ripped away. And in the lungs, it's going to be more receptive to the oxygen, and once bound, it'll hold on to it pretty tight. In the tissues, carbon dioxide flows into the red blood cells, and when it binds to the hemoglobin, it promotes a change in shape that promotes the release of oxygen from a different binding site, right? So as CO2 goes in, oxygen comes out in the tissues, okay? The oxygen attaches to things like amino groups in the hemoglobin that are different from the oxygen binding site and changes the shape of the hemoglobin, makes it less likely to grip the oxygen very well. And then the other thing that the CO2 does is it forms carbonic acid, all right? And when the carbonic acid forms, that's H2CO3, what happens is that the bicarbonate, which is the HCO3 minus, leaves the red blood cell and sits around in the plasma, while the protons are going to bind to the hemoglobin and further reduce its capability of holding on to its oxygen, okay? And so those are called the Haldane and the Bohr effect, okay? So that's not going to happen in the lungs, right? Because CO2 levels and pH levels are flipped, right? CO2 levels are low in the lungs, and pH levels are not acidic, okay? So the hemoglobin is going to tend to soak things up. Hematopoiesis takes place in red bone marrow, which is in the bones of the axial skeleton and the proximal femur and humerus and the pectoral and pelvic girdle, okay? So what happens here is that hematopoietic stem cells give rise to all of the other formed elements of blood, okay? What turns them towards a certain path, a certain mechanism to create the blood, is the colony-forming factors, which are proteins that bind to receptors on these stem cells and then guide them down a certain developmental pathway in an irreversible fashion, okay? Once the cells begin to change their characteristics, then they can't go back the other way, okay? It's like going through a series of one-way doors, and once you've done that, there's no way of going back to a previous state, okay? So this change in characteristics through time is called differentiation, all right? And there is no de-differentiation that we see in red blood cells. So what you're looking at here is how we make a red blood cell. This is erythropoiesis, which is a branch of hematopoiesis, all right? Myeloid stem cells transform into proerythroblasts in about 2 weeks. You can see those here, okay? The proerythroblasts develop into basophilic, polychromatic, and orthochromatic erythroblasts. So basophilic would look purplish blue, right? Polychromatic would have multiple colors, and orthochromatic would have a single color, all right? And then what happens is that we form a reticulocyte after we eject the nucleus from the erythroblast, right? So the erythrocyte is an anucleate precursor to the erythrocyte, okay? So your reticulocyte count in your blood is an indication of your rate of hematopoiesis. The more reticulocytes you have, the higher your rate of hematopoiesis, okay? How do we regulate this? Well, if you got too few red blood cells, you're going to have a condition called hypoxia, in which there's low oxygen concentration in cells and tissues that can kill them. Too many red blood cells is going to cause the blood viscosity and the blood pressure to go way up, and you're going to have reduced flow as a result of that, and that's going to cause problems in terms of increased risk of the formation of emboli and increased risk of blowing capillary beds. That can increase the risk of heart attack and stroke. There's about 2 million red cells made per second, and the balance between production and destruction is hormone dependent and building block dependent. Okay, the main hormone here is erythropoietin, which is made by the kidneys. Okay, and it responds to low oxygen concentration in the blood. The EPO flows through the blood, gets to the red marrow, and then hits those myeloid stem cells, and then cranks up the rate of red blood cell production. Okay, one of the effects of testosterone, for instance, is to jack up erythropoietin levels. That's why testosterone jacks your crit. Okay. You also need the components that make up a red blood cell. So you need iron, you need all the amino acids, and you need B vitamins because those are used to make DNA. And remember that DNA is present in the precursors, right? All these guys are whipping out a lot of DNA, and we don't dump the nucleus until we form the reticulocyte. So the B12 is critical for that. And remember that B12 is absorbed across the lining of the stomach using a protein called intrinsic factor. Okay, and that's what gets it into the bloodstream and then gets it to the red marrow. If you don't have intrinsic factor, or you make too little of it, you end up with a condition called pernicious anemia, meaning you got to get B12 shots in order to maintain your red blood cell levels. Okay, so that's erythropoietin, right? High red cell or low oxygen levels are going to reduce production, and the reverse is going to increase production. Okay, hypoxia that is sensed by the kidneys and the liver is going to jack up your rate of erythropoiesis. Okay, causes of hypoxia: you can have decreased red numbers due to hemorrhage or increased destruction. You can have insufficient hemoglobin per red blood cell, such as in iron deficiency anemia, and you can have reduced oxygen availability, such as in high altitudes. Okay, your body, in response to these kinds of situations, is going to jack up your hematocrit to promote your survival, and this is a normal response, right? Here you have a lack of building blocks, okay, and here you have a problem with the marrow or with the turnover. Okay, the marrow might be impeded, or you might be losing blood internally or externally. So erythropoietin causes rapid maturation of committed marrow cells and increased circulating reticulocyte count in about 2 days. And you might remember the name Lance Armstrong. Lance was the young man that won all those Tour de France's, and it turns out part of the reason he won all those things was because he was cheating. Okay, what he was doing was he was using erythropoietin through a needle to jack up his crit, and that increased his endurance. And the result was he was able to beat the pants off the people that weren't using. Okay, it turns out though that there were other people on the tour that were using too. Lance just took the blunt force to his career. All right, now he's done. Testosterone enhances erythropoietin production, and that's why the male has a higher crit. Okay, so here's how it works, right? Hypoxia, right, causes decreased red count, decreased oxygen, decreased hemoglobin. The kidney picks this up, kicks out EPO, goes into blood to the red marrow. Here we're showing you a proximal femur, and then the red cell count goes up because the red cell production goes up, and now we're back where we need to be. Okay, that condition of low oxygen that causes an increase in hematocrit above what you see in people at sea level is called secondary polycythemia. Okay, secondary because It's produced as a result of a different phenomenon, in that case, living at high altitudes where there's thin air, okay? And polycythemia just refers to the excessive number of red blood cells, okay? What do we need to make blood? Well, we need amino acids, lipids, and carbs. We need iron. We get all this from our diet. 65% of our iron is in our hemoglobin. The rest is in our liver, spleen, and bone marrow. Oddly enough, those are all blood-rich organs as well, okay? Free iron ions are toxic, and so they have to be stored inside proteins such as ferritin and hemosiderin, or in the case of blood cells, hemoglobin, okay? There's another reason that you have to store iron like that, which is that the solubility of iron ions in water is very low, which means that they like to form solids. They like to precipitate. So if you have a high concentration of iron ions in an aqueous solution, you run the danger of basically generating iron oxide, rust, inside your body. And you don't want that to happen, so you connect it to protein, and that avoids that problem, okay? It's transported bound to transferrin in the blood, right? It's stored in the cells in the hemosiderin or the ferritin. B12, we've talked about why you need that, okay? And now let's talk about the life of a red blood cell, right? 120 days. Why so short? No protein synthesis, no growth, no cell division. Old red cells get fragile. Hemoglobin degenerates. It gets trapped in circulatory channels, especially in the spleen. The macrophages eat the dying red cells in the spleen. The heme and the globin separate. The iron is then salvaged to be reused, okay, because the body wants to take advantage of everything it's got. The heme is degraded into a pigment called bilirubin, which ends up in the bile as bile salts, and that's why bile is green, okay? That goes into the intestines. It emulsifies fat in our diet there, and then when it gets to our colon, it becomes stercobilin, which is brown, and that's because of the action of the bacteria that live in our colon on the bilirubin, okay? Another pigment, urobilinogen, is excreted in the kidneys, and that's part of what makes urine yellow, okay? The globin gets metabolized into amino acids, and that gets released into circulation, and that protein gets reused. Anemia is just low oxygen carrying capacity in the blood, okay? It's usually a sign of a different condition rather than the condition on its own, all right? Blood oxygen levels can't support normal metabolism, so often what we see is fatigue, pallor, shortness of breath, and chills in individuals that have anemia, right? Three groups of anemia include blood loss anemia, low red blood cell production anemia, and high red blood cell destruction anemia. All of these result in a lower crit. And if your crit's low, your oxygen carrying capacity is low, and that means that your tissues aren't going to get what they need in terms of oxygen levels to continue to produce enough ATP to function. Because remember that in the presence of oxygen, you make 20 times more ATP than without it on average, okay? Hemorrhagic anemia is the result of blood loss, such as through a stab wound. We treat it by blood replacement. Chronic hemorrhagic anemia is a persistent blood loss through things like hemorrhoids or bleeding ulcers, so internal bleeding. You treat the primary problem and you deal with it. In hemorrhagic anemia, we do sealing of the wound, right? And then we do replacement through transfusion of the correct blood type, obviously. Iron deficiency anemia can be the result either of a lack of iron in the diet or an inability to absorb iron into the blood, okay? You end up in iron deficiency anemia with what we call microcytic or hypochromic red cells. Microcytic just means they're tiny. Hypochromic means they lack color. Okay, and we use iron supplements to treat them. Pernicious anemia is an autoimmune disease that destroys the mucosa of the stomach, and the result is that we don't have intrinsic factor to escort vitamin B12, which is a water-soluble vitamin, across the lining of the stomach and into the bloodstream. The red cells can't divide, and we form macrocytes, and we treat these with B12 injections or a B12 nasal gel. Right? It can also be caused by low B12 in the diet, right? And this is a danger in people that are vegan or vegetarian because a lot of our B12 comes from animal sources. Renal anemia is a lack of erythropoietin. This often accompanies renal disease and is treated with synthetic erythropoietin. While aplastic anemia is the result of destruction or inhibition of red marrow by drugs, chemicals, radiation, or viruses. So this is one of the reasons, for instance, that somebody who's under chemotherapy or radiation treatment becomes anemic and gets very sick, because we're attacking, in those cases, cell division, and cell division is a major part of blood production. Okay, often the cause is unknown. Okay, anemia of this type can also result in clotting and immunity defects. Well, that makes sense, right? Because if we're hitting the marrow full scale, you're also going to cut down on white cell production, okay? And also with platelet production. We treat it short term with transfusions, okay? And long term, we can do something called bone marrow replacement, okay? So this is kind of scary, because what you do here is you take somebody and you kill all of their red marrow with something like radiation. And then you give them new marrow from a compatible donor, and then that repopulates those red marrow spaces, and then that person has a new source of blood production, okay? But this is dangerous, because if we kill all your red marrow and then the transplant doesn't take, then you're dead, because you have no source anymore of blood, right? So that's a disaster. So you're really, going out on a limb when you have to go to bone marrow transplant, right? All right, let's take 15 minutes, and then we'll come back at let's say okay, guys. We're going to finish up chapter 17 here, talking about blood and its functions. And we had actually left off talking about hemostasis. And what you're looking at here is a step-by-step breakdown of what happens when the body attempts to restrict the flow of blood out of the body in order to maintain now all the valuable components that you find in both the formed elements and in the plasma. So step one is a vascular spasm. Now, what does that mean? That means that the smooth muscle, which is found primarily in the tunica media of the blood vessels, that's the middle layer of tissue that's bordered on the outside by the tunica externa and the tunica intima, what it'll do is it'll contract. And it does this in response to the production of certain chemical intermediates that leak out of the cells during tissue damage. The next thing that will happen is that blood flow will temporarily stop through that region, and this helps to decrease the amount of blood loss at the point of the injury. The next thing that will happen is that platelets, also known as thrombocytes, which you find as the second most numerous element in the formed elements of blood, will snag on the damaged tissue and will begin to burst and release clotting factors. So as they adhere to the collagen fibers and begin to release their clotting factors, they also begin to form what we call a platelet plug. So this is like a temporary block to keep us from losing more blood than we otherwise would. And then as the clotting factors begin their cascade, what happens is that we begin to form now an actual blood clot, and this is what's called coagulation. Coagulation ends at the point at which fibrinogen is converted into fibrin, which forms a mesh, and this mesh traps red blood cells and platelets, and this becomes now the second plug at the site of the injury. What happens afterwards is that cells will move in and attempt to repair the injury, and then eventually the clot will be dissolved after it's served its useful purpose. So if we look at these processes in a little bit more detail, and we examine coagulation, prothrombin activator is formed in both intrinsic and extrinsic pathways. What are these pathways? They're just two different chemical cascades that end up in the production of a blood clot, and they actually start out responding to completely different stimuli, but they end up ultimately as the conversion of fibrinogen to fibrin and the production of the clot. Prothrombin is converted to the enzyme thrombin, and thrombin is what actually catalyzes the conversion of fibrinogen into fibrin, and it's the fibrin that's the insoluble protein that ends up trapping the platelets and the RBCs and whatever else happens to wander by the injury site. So let's examine these two pathways that lead to prothrombin activator. It can be initiated by either the intrinsic or extrinsic pathway, but often by both at the same time. It's triggered by tissue-damaging events, which involves a series of procoagulants. Well, what the heck is a procoagulant? It's just a chemical that promotes coagulation. Each pathway cascades towards a protein that's in the plasma known as factor 10. Factor 10 complexes with calcium, PF3, and factor 5 to form prothrombin activator. Prothrombin activator is what is going to eventually trigger the production of fibrinogen into fibrin. The intrinsic pathway is triggered by negatively charged surfaces, such as activation by platelets, collagen, or foreign substances. It uses factors present within the blood already, while the extrinsic pathway is triggered by exposure to tissue factor or factor 3, which is an extrinsic factor. So the starting points are a little bit different. When you're looking at the intrinsic factor, we're talking here about something that's coming in from the outside, basically. Exposure to collagen, for instance, would result from injury to tissue. Activated platelets would result from them basically sticking to the collagen fibrils. Glass and foreign substances often are seen at the site of an injury, such as in a laceration or in a puncture. So it uses factors that are present within the blood. That's why it's called intrinsic pathway. The extrinsic pathway is triggered by exposure to tissue factor or factor 3. It bypasses several steps of the intrinsic pathway, so it ends up actually being a quicker way to generate a clot. Prothrombin activator catalyzes the transformation of prothrombin to the active enzyme thrombin. Whenever you see the prefix pro in front of the name of a particular enzyme or protein, what it means is that it's in an inactive state. And generally what happens is that it'll be transformed, usually through an enzymatic cleavage, from an inactive to an active state, at which point now it is able to either catalyze the chemical reaction in which it's involved, or, in this case, to trigger the clotting cascade. Once prothrombin activator is formed, the clot forms in about 10 to 15 seconds. Now you might wonder why it is that prothrombin is produced in an inactive form initially. And the reason is so that clotting doesn't spontaneously occur all over the body. The body uses this strategy when it wants to delay the activation of a particular enzyme or protein in a cascade so that it doesn't just begin to spontaneously fire, spontaneously execute. The idea behind the prothrombin is that it's there if it's needed, but we need an extraordinary event in order to trigger its activation. So as produced straight off of the messenger RNA, this protein isn't capable of doing anything. It's only after cleavage of particular bonds between amino acids, that it goes from the inactive to the activated form. Okay, so now we get to the common pathway. Thrombin converts fibrinogen into fibrin, which strands form the basis of a clot by trapping now passing platelets, passing erythrocytes. It also causes the plasma to become a gel-like trap, and this further ensnares the formed elements. The thrombin, along with calcium, activates factor 13, which cross-links the fibrin and strengthens and stabilizes the clot. And then that clot is going to stay around until it's no longer needed, at which point clot-busting enzymes will dissolve the clot. The components of the clot will be recycled, and by that time what we've done is we've effected tissue repair so that we quit the loss of blood. And so here you're looking at a diagram showing you these different parts of the clotting pathway. So in phase 1, let's look, for instance, at the extrinsic pathway first. Okay, this is where we're starting the whole thing. We have an injury, right? A tissue cell trauma exposes the blood to tissue factor. Why? Because we've damaged cells in the area and they're starting to leak tissue factor into the surrounding environment. Tissue factor, along with calcium, along with factor 7, eventually ends up forming a tissue factor, factor 7A complex, and this is going to catalyze the conversion of factor 10 into an activated factor 10, which is ultimately going to activate factor 5, and then factor 5 is going to, along with calcium and PF3, activate prothrombin. Okay, so that's the extrinsic pathway. And you might say, well, where are all these crazy factors? Where are they? They're in the plasma. They're already there. They're already made, and they're just kind of waiting around for an event. Okay, in the intrinsic pathway, what we have is the triggering event being the vessel endothelia rupturing and exposing underlying tissue such as collagen. Okay, so again, an event from the outside has disturbed our homeostasis. The platelets cling to their surface and provide sites for mobilization of the different factors. We start here with the activation of factor 12, which in turn activates factor 11, which in turn, along with activated factor 7, activates factor 9, with the presence of calcium being very important in this pathway, and then along with PF3, which is released by aggregated platelets rather, we activate now factor 8, and we form now an activated factor 8 and 9 complex, which activates factor 10, which activates factor 5, which ultimately, in the presence of calcium and PF3, activates prothrombin. And so if you look at this, you might think, well, gee, that's awfully complicated. And you'd be correct. That's several steps. If we look at the intrinsic pathway, you've got here 1, 2, 3, 4, 5, 6, 7 steps. Why so many steps? Why can't we just do it all in one step? Well, the reason for this is that, remember, that a cascade is set up to amplify a biological signal. That's the whole strategy behind this arrangement of proteins that are waiting to be activated. And remember, we talked about using the illustration of freeze tag, a game you used to play when you were little, right? If you were it in freeze tag, what did you get to do? You got to run around, and everybody you touched, they had to freeze and not move. And you could touch more than one person. Okay? And it's the same sort of strategy here with the intrinsic pathway. At each step in the cascade, it's not as if it's just a one-to-one correspondence. It's not as if, for instance, when we activate factor 12, it only activates one molecule of factor 11. It may activate several or several hundred. In turn, factor 11 may activate several hundred factor 9s and so forth. And so what happens in this cascade is that the signal gets amplified at every step. It gets bigger and bigger and bigger and bigger and bigger, okay, until we finally end up ultimately with the production here of a clot. Now, not only do we amplify the signal at each step in the cascade, and as a result respond very rapidly to the stimulus, the other nice thing about a cascade is the cascade's easy to shut off. Because all you have to do to shut the cascade off is to stop now the initial event. Okay? In the case of the intrinsic pathway, this would be now the repair of the damaged vessel wall, right? And now no longer will we be activating, in this case, factor 12. In the case of the extrinsic pathway, it'll happen once the tissue factor is no longer being secreted into the blood plasma, and at that point, this entire cascade will stop. Okay, so that's the whole point behind having this complex pathway that gets you to a single endpoint: the amplification of the signal and the ability to very easily turn the cascade off once it's served its purpose. And we see cascades in living systems in a lot of different places. We see them, in this case, in an extracellular cascade, and we see them also inside cells when we look at things like receptor signaling, which involves a sequence of events that ultimately ends up generally with either the transcription of a particular gene, which results in the production of a gene product that carries out a particular task, or perhaps the opening of an ion channel or some other immediate response by the cell. At which point, now that we've carried out the task of the cascade, we can turn off the initial events that started it and then return to our original state. Now, if we look at phase 2, what we see here is prothrombin being activated in becoming now thrombin. And the thrombin, the activated thrombin, is able to convert fibrinogen, which is, again, we can call it the inactive form of fibrin, if you like, because it's not capable of forming a clot on its own. It's soluble in plasma. But once thrombin acts on it, what happens is it cleaves particular covalent bonds in the fibrinogen, and now this becomes an insoluble protein that is capable of trapping cells as they pass by, and it also, all by itself, is an insoluble polymer. Okay, this reaction is also dependent on the activation of factor 13, which happens in the presence of calcium and thrombin, and this ultimately results now in this cross-linked mesh in which we're capable of trapping now all manner of formed elements, and this is going to serve now as our temporary block against blood loss while other cells come in and repair the tissue. So let's talk about what happens after we make the clot. Once we've got the clot in place, that's sort of like on a construction project putting up the do not enter sign, say on a highway entrance ramp, or a store closed sign, right, while the crew is in there effecting repairs. So once the clot is up, that's like our do not enter sign. Now what happens inside the tissue that has been damaged? What's going on? Well, clot retraction is basically where the clot begins to shrink. The clot will stabilize. Actin and myosin in the platelets will contract within an hour to half an hour. And this pulls on the fibrin strands, and it squeezes serum out of the clot, and it draws ruptured blood vessel edges closer together. And what's the purpose of that? Well, to make the repair easier, right? It's much easier to stitch two things together if they're near each other than if they're really, really far apart. So let's talk about vessel repair. Once we've brought the two ends in close apposition, the vessel can now affect healing. Platelet-derived growth factor is a chemical signal that stimulates the division of smooth muscle cells and fibroblasts, and this helps rebuild the blood vessel wall. And vascular endothelial growth factor stimulates the endothelial cells to multiply and restore the endothelial lining. So basically what's happening here is that chemical signals are being given off that promote the multiplication and the growth of the cells that are eventually going to seal off the wound and perform the same function as the original tissue. Fibrinolysis is what takes place after we've repaired the tissue. We can now get rid of the clot because we've effected our repairs. So it removes the unneeded clot after the healing takes place. This usually begins within 2 days and continues for several days afterwards. Plasminogen in the clot is converted to plasmin by tissue plasminogen activator, abbreviated TPA, factor 12, and thrombin. Plasmin is the fibrin-digesting enzyme that results from this series of events. Two mechanisms can limit clot size because we don't want clotting to run out of control. In fact, there are conditions in which clotting happens in an uncontrolled fashion, and this results in all sorts of problems. If you start to spontaneously clot throughout the circulatory system, you can imagine some of the problems that might occur, right? If you think about it. Now think for a minute, what would happen if your blood just started to spontaneously clot all over your body? Well, eventually those clots, if they were moving clots, which are called emboli, could lodge in capillary beds and choke off the flow of blood to downstream tissue and cause it to die, right? That'd be a disaster. That would result in tissue necrosis, cell death. Other things that would happen is that with these formation of all these spontaneous clots all over the place, you would actually begin to use up a lot of the components of the clotting cascade, and paradoxically, if you were injured, you would begin to bleed and you'd be unable to stop the blood loss because, again, you've run out of all of those factors that have to be present in a certain concentration in order for the clotting cascade to execute. Okay, so two mechanisms limit clot size. Swift removal and dilution of the clotting factors is one. Inhibition of the activated clotting factors is another. Now this is interesting, okay, this swift removal and dilution of the clotting factors. This happens because your blood flows through your blood vessels, okay? And how does the blood flow through the blood vessels? Primarily through the pumping action of the ventricles of the heart, but also to some extent by the fact that you move, right? One of the things we're going to find out when we talk about the blood vessels this coming Saturday is that in the arteries, blood moves around primarily because it's close to the pump, the heart. But once we get to the capillaries, one of the things that happens is that the pressure in those vessels drops dramatically because of all the resistance that the blood meets as it tries to flow through a very narrow channel. And then when the blood is recollected by the veins, the pressure is so low that we have to have some other mechanism to get it back up to the heart. And it turns out that that mechanism involves the valves that you find in veins that are what we call one-way valves that eventually work the blood back up to the heart as you move, as you walk, as you stand, sit, and run, and so forth. By squeezing the blood up through a series of one-way valves, ultimately to get back to the heart through the right ventricle as it arrives there from the superior and inferior vena cava. So depending on whether we're talking about the arterial system or the venous return to the heart, you've got slightly different factors that move the blood around. But the point is that the blood moves, okay? So right here, okay, this very first point, swift removal and dilution of the clotting factors keep the clotting from running out of control, right? That's good. But on the other hand, think about what happens if blood flow slows down, okay? How can blood flow slow down? Well, you could have, say, a really bad injury, okay? And because of inflammation and swelling, you could inhibit the flow of blood in and around the injury. Well, what could this result in? This could result in the production of emboli, right? And we already know what emboli can do. This is also a problem in individuals that suffer from a condition known as varicose veins. Varicose veins are a distension of the venous system, particularly in the lower extremities. And you find them primarily in people that perform jobs where they have to stand for long periods of time and the blood tends to pool, because you have to remember that it's tough to get blood back to the heart from below the heart because you're fighting gravity. It's easy to get blood back to the heart above it because gravity is working with you. And so if you stand and have to remain relatively immobile for long periods of time, what happens now is that blood flow in the venous system slows and you can form spontaneous clots. And so that's one of the reasons that you always want to move a little bit, even if you're seated or you're standing, because you don't want that blood to pool. You don't want spontaneous clotting to happen. It's also one of the reasons why you elevate affected limbs when you're attempting to heal from either a surgical procedure or a bad injury, because we don't want that spontaneous clotting to occur. The inhibition of activated clotting factors is another limiting factor in the growth of clots. Thrombin bound onto fibrin threads is also a limiting factor as well. And antithrombin 3 is a protein that inactivates unbound thrombin, and as a result now keeps it from executing now the clotting cascade outside of its normal boundaries. Heparin, present in basophils and mast cells, also inhibits thrombin by enhancing antithrombin 3. Heparin is a member of a class of drugs called anticoagulants. And basically the way these components work is that they inhibit the clotting cascade and they decrease blood viscosity. If you've ever seen a package of Decon in the grocery store, Decon is a it's a chemical that is impregnated into feed for mice, and you put this in the drawers in your kitchen and wherever you have a mouse, rat, or squirrel problem. And what happens is that the Decon contains a component known as warfarin, which is an anticoagulant. And what happens is that these little animals get a heavy dose of this anticoagulant, and then they go off and do what rodents do, which is get in fights and puncture and lacerate themselves, and they can't stop bleeding because of all the anticoagulant that's in their blood. And the idea is that the way the Decon operates is that the mouse will be far, far away from your house by the time that happens, and so he'll bleed to death out in the yard somewhere as opposed to dying inside your walls, as would happen with a mouse trap. And as a result, this is one of the things that you read on the Decon package, is that you don't want your dog, your cat, or your children to be near any of that stuff or to consume it, because it'll have the same effect on them. But heparin and warfarin are both anticoagulants. Okay, platelet adhesion is prevented by the smooth endothelia of the blood vessels that prevents the plates from clinging. Basically, the endothelia normally repels the formed elements of blood, keeps them from sticking there. There's also antithrombic substances such as nitric oxide and prostacyclin that are secreted by endothelial cells. And vitamin E acts as a potent anticoagulant as well. There are sometimes when this clotting cascade doesn't work the way it's supposed to. These are called disorders of hemostasis, and you can probably guess what two classes they fall into, either too much clotting or too little clotting. Okay, and so within these two broad classes, thromboembolic disorders are undesirable clot formations, while bleeding disorders are abnormalities that prevent normal clot formation. Disseminated intravascular coagulation involves both types of these disorders, and essentially what happens in this disease is that you spontaneously clot throughout the body. And we already talked about the disastrous consequences of this. The fact that you titrate out, you dilute those clotting factors to the point where if you do have an injury, you'll tend to bleed out, and then you produce all these emboli that travel around the body and can lodge very easily in capillary beds. Now you might think, well, gee, big fat deal. I've got an embolus lodged in a capillary bed, and some tissue might die. Why is this a big deal? Well, generally speaking, it's unpleasant but not life-threatening. But if it happens in the heart, it can cause a heart attack, and if it happens in the brain, it can cause a stroke. And both of these are obviously lethal outcomes in many cases. A thrombus is a clot that develops and persists in an unbroken blood vessel, and it can block circulation, leading to tissue death. And this is that first kind of tissue death we talked about in class, necrosis. This is tissue death due to insult or injury. An embolus is a thrombus that is freely floating in the bloodstream, so it's on the move. And it eventually will lodge in a narrow vessel. Okay, basically the diameter of the blood vessels decrease and decrease and decrease until we get all the way to the capillaries, which are the narrowest vessels in the body. It's very difficult for a clot to traverse a tiny little capillary. It'll easily lodge there. An embolism is an embolus obstructing a vessel, such as a pulmonary or cerebral embolus. Pulmonary embolus would be in the lungs, for instance, and a cerebral embolus would be in the brain tissue. What are some of the risk factors for these thromboembolic conditions? Well, arteriosclerosis, inflammation, slowly flowing blood, or blood stasis from immobility. So let's break down each of these. Arteriosclerosis, what the heck is that? I'm sure you've heard that term before, but you've never really examined what it is. Basically, arteriosclerosis is a narrowing of the blood vessels due to the deposition of plaque on the surface of the endothelium. So what happens in arteriosclerosis is that the vessels become hardened and the lumens begin to narrow, and as a result, the pressure in those vessels goes up. The danger here is that as the pressure in these vessels goes up, two things can happen. You can burst capillary beds by increasing the systemic pressure, because there's only a certain amount of pressure that the capillary can withstand before it'll blow. And if that happens, what you'll have is tissue death due to lack of blood flow, not because of the block of that capillary, but because now we're bleeding out. Okay, and so we're not getting the blood to all the tissues in a timely fashion. The other problem that can occur as a result of these narrowed lumens is that the plaque itself can get knocked free from the endothelium, and it by itself can act as an embolus that can lodge in a capillary bed. And obviously, we talked about the fact that this is going to increase the risk of myocardial infarction, aka heart attack, or a stroke. How about inflammation? What is inflammation? Well, inflammation is just the body's response to injury, and the classic signs of inflammation include edema, erythema, pain, and increased temperature. Okay? Now, edema, what the heck is that? Edema just means swelling up, okay, usually due to an accumulation of fluid. Erythema means reddening, and this is usually the result of blood flowing to the area. Pain, you know, okay, and the increased heat is a function partly of the blood coming to the area, but partly as a result of chemicals that are released at the site of the inflammation that are designed to increase the temperature in that particular tissue compartment. The inflammatory process is a good thing, generally speaking, because it is designed to deal with an injury or an infection by bringing blood to the area, and with that blood come the white blood cells that are going to fight the infection and repair the tissue. But if we have generalized inflammation inside the vessels or in tissue, what can happen now is that we're going to slow the flow of blood, and as the blood flow slows down, now we're going to spontaneously clot, because what will happen is that those clotting factors will become transiently concentrated, and if they become too concentrated, the clotting cascade will just randomly fire. Okay? Other risk factors include slowly flowing blood or blood stasis from immobility. Slowly flowing blood, how can that happen? Well, inactivity is one way that can happen, right? If you don't move much, then you're not going to be able to get a lot of venous return to the heart, because you're not doing a lot of skeletal muscle contraction, which is what pushes the blood back to the heart from the lower extremities in the fight against gravity. Okay? Slowly flowing blood can also result from a healing injury. You can imagine, for instance, somebody that's had a surgical procedure done, and the place where the surgery has been done has a lot of edema, okay? And if there is a lot of tissue fluid there's a lot of pressure on the vessels. There's a lot of pressure on the vessels. You're going to slow the flow of blood through the area, and spontaneous clotting can be the result. So let's talk about some common anticoagulants. Aspirin is an easy one. It's an antiprostaglandin that inhibits thromboxane A2. Heparin is an anticoagulant used clinically for pre- and post-operative cardiac care. Warfarin is the component that we talked about in decon, also known as coumadin, and it's used for those prone to atrial fib. It interferes with the action of vitamin K, and as a result, now um, decreases blood viscosity and the tendency of the blood to clot. Dabigatran directly inhibits thrombin. Okay, so all these are anticoagulants, and they work in a slightly different at a slightly different point in the coagulation cascade, but the end product is the same. The blood tends not to clot. Okay. Now you might say, well, wait a minute, vitamin K. Where did vitamin K come in? Well, vitamin K is one of the fat-soluble vitamins, A, D, E, and K, and it's actually produced in the large intestine by bacteria that ferment the fiber and the other waste products that come from the small intestine into the large intestine. We absorb this vitamin K directly into the bloodstream, and it serves as a cofactor in the clotting cascade. Okay, so we need vitamin K either as a result of our endosymbionts, the bacteria that live with us, or we need to ingest it as a supplement in order for clotting to occur. Okay, let's talk about bleeding disorders. Thrombocytopenia is a deficient number of circulating platelets. Petechiae appear due to spontaneous widespread hemorrhaging. And what are petechiae? They're just little spots of blood where you sort of bleed out under the skin, and they look like little red splotches. You can see them in the sclera of the eye. You can see them under the skin, and they indicate now that we have a deficiency in the clotting cascade. It's usually due to a suppression or a destruction of red marrow, which can come from either a malignancy in the hematopoietic stem cells, so cancer of the hematopoietic stem cells, can come from radiation treatment, and it can also come as a result of certain drugs. A platelet count below 50,000 per microliter is usually the diagnostic tipping point, indicating thrombocytopenia. It's generally treated with transfusion of concentrated platelets. Impaired liver function can also lead to bleeding disorders because the liver produces procoagulants. Causes include vitamin K deficiency, hepatitis, and cirrhosis. Cirrhosis is essentially the death of liver tissue, and as a result, the loss of liver function, often due to abusing recreational drugs or excessive intake of alcohol over an extended period of time. We can also have impaired fat absorption, and liver disease can also prevent liver from producing bile, which impairs fat and vitamin K absorption. So even though the vitamin K may be around, if the liver isn't doing its job, we're going to have a difficult time getting it ultimately into the circulation from the small intestine, and as a result now we're going to lack this critical component in the clotting cascade. I mean, you have to think now in terms of what's important, right? If you have a multi-step process, it's good in the sense that the way that the cascade process is set up, we magnify the initial stimulus and ultimately end up at our goal, in this case of creating a clot. And it's also good in the sense that we can easily turn it off once it's served its purpose. But it's bad in the sense that there are many opportunities to have a component go missing, right? And if any of these components in the clotting cascade is missing or deficient, you're not going to clot, and you're going to tend to bleed out. Okay? Hemophilia is another type of bleeding disorder. It includes several similar hereditary bleeding disorders. Hemophilia A is the most common type, makes up a little over 3/4 of all cases, and it's a factor VIII deficiency that's inherited. Hemophilia B is a factor IX deficiency, while hemophilia C is a mild type, and it's a factor XI deficiency. Symptoms include prolonged bleeding, especially into joint cavities. We treat this with plasma transfusion and the injection of missing factors. What are some of the risks here? Increased risk of hepatitis and increased risk of HIV. What is hepatitis? It's an inflammation of the liver, often due to a viral infection. And HIV stands for human immunodeficiency virus, and this is the agent of AIDS, which is the acronym for acquired immune deficiency syndrome. And essentially what happens here is that we leave ourselves open now for viral attack as a result of having these problems with these disorders, these bleeding deficiencies. Now, several of these hemophilias, such as hemophilia A, are found, their genes are found on the X chromosome, so they are said to be sex-linked. Sex-linked trait is a trait that occurs on the X. You have to remember that women are born with 2 X chromosomes, while men are born only with 1 X chromosome. And so for men, anything that's defective that shows up on the X is going to be expressed as a trait. So men are said to be hemizygous for the genes that are present on the X. And so a lot of conditions that are essentially recessive conditions, where normally you'd need 2 bad genes in order for the disease state to show up, in men you only need 1 because the Y chromosome has no genetic information to cover up what is being expressed on the defective gene, whereas women with 2 X chromosomes would need to have 2 bad copies of a defective gene in order for the disease to be expressed. So there are several sex-linked traits that are more prevalent in males and females. Hemophilia is one, color blindness is another, and male pattern baldness is a third. Okay, disseminated intravascular coagulation is widespread clotting that causes bleeding and also tissue necrosis due to lack of blood flow to certain areas as capillary beds get blocked due to these spontaneous clots. Widespread clotting blocks intact blood vessels. Severe bleeding occurs because residual blood is unable to clot because all those clotting factors have been used up in producing these spontaneous clots. It can occur as a pregnancy complication. It can also occur in septicemia. Now, what is septicemia? That's just the presence of bacteria in the bloodstream. It's a very dangerous condition because bacteria in the bloodstream can travel now all over the body and infect all the internal organs. And this can lead to a condition called MODS. Let's draw that up here. MODS. And what does MODS stand for? It stands for multiple organ dysfunction syndrome. And this can very often lead to death. Okay? So this is one of the reasons why, and you'll see this when we talk about the immune system, the body has so many fail-safes in place to try and keep bacteria from getting into our tissue fluid or into our bloodstream, because if it does, we have a terrible time fighting it off. Transfusions are basically the movement of blood from a donor to a recipient. Whole blood transfusions are used when blood loss is rapid and substantial. Packed red cells, which are plasma and white blood cells which have been removed, and the packed red cells are usually suspended in physiological saline, are transferred to restore oxygen carrying capacity in the blood. So when you donate blood, for those that are from Cincinnati, the place that does that is Hawksworth in Dayton. I'm sure there is a major facility there where they take blood donations. What they do when they take a unit of whole blood is that they essentially bleed you out into a plastic bag. They take the whole blood, and then they'll take that unit and they will rinse the plasma away using physiological saline. And what this does is this dilutes out your antibodies and your plasma proteins while preserving the red cells and keeping them from bursting, because what they're suspended in now is an isotonic salt solution. And so this alleviates any problem that you might have with the donor antibodies and the recipient antigens when you're attempting to give a transfusion. This is typical procedure for blood donations. So we don't want those white cells, and we don't want those antibodies in there. They could cause problems. Transfusion of incompatible blood can be fatal because of a process known as agglutination. And we saw this in lab last Saturday when we did our ABO testing. Agglutination is just the clumping together of red blood cells. Okay, so let's draw a couple of red blood cells here. And this occurs as a result of the interaction of red blood cells with antibodies. And antibodies have a minimum of 2 antigen binding sites on their surface. And so what I'm drawing here, this little Y-shaped thing, that's the antibody. This is the RBC. Let's fill it in. Okay. And so what you can see happening here as this complex begins to grow is that eventually we're going to form this high molecular weight complex that will eventually fall out of solution. And eventually these red blood cells will be destroyed in a process called hemolysis. Okay, this is a bad deal. Okay, these damaged blood cells, number one, can't carry oxygen. Number two, it can result now in the production of emboli that can end up now blocking capillary beds and killing the recipients. This is why blood typing is always done prior to a transfusion in order to determine the compatibility of the recipient for the donor blood. Red blood cell membranes bear about 30 different types of glycoprotein antigens. Okay, and here we go again, glycoprotein. What is a glycoprotein? Okay, glyco means sugar. And protein means protein. Okay, so what is a glycoprotein? It is a protein with sugar components attached to it. And these antigens act as ID tags for your red blood cells. So anything perceived as foreign can generate an immune response. And what would be perceived as foreign is anything that doesn't have the same antigen display as your normal tissue. Promoters of agglutination, called agglutinogens, are what we refer to these red blood cell antigens as. Mismatched transfused blood can be perceived as foreign. It can be agglutinated and destroyed, and this can be fatal. The presence or absence of each antigen is used to classify blood cells into different groups. Antigens of the ABO and RH blood groups cause vigorous transfusion reactions. Other blood groups, such as MNS, Duffy, Kell, and Lewis, are usually weak agglutinogens, and they don't come into play when we're considering transfusion compatibility. But essentially, the bottom line to remember here is that whatever antigen you're born with on your red blood cells, you are not going to have the antibody for in your serum, because if you did, you would agglutinate your own blood and you would die. But you will have antibodies against all the other antigens depending on your blood type. Okay, so let's look at the ABO blood groups first. The different types of blood groups with the ABO system are A, B, AB, and O. And it's based on the presence or absence of 2 agglutinogens, A and B, on the surfaces of red blood cells. Blood can contain preformed anti-A or anti-B antibodies known as glutinins. Okay, so a glutenin will react with an agglutinogen to form an agglutination. Okay, so those are 3 words that you want to make sure that you have clear in your mind. They act against transfused red blood cells with ABO antigens not present on the recipient's red cells. Anti-A or anti-B form in blood at about 2 months of age, and they're at adult levels by age 8 to 10. And what you're looking at here is just a chart showing you the ABO groups and what molecules are present in each of the different ABO blood types. Okay, so let's start with an easy blood group. Let's start with type O blood. Okay, an individual with type O blood does not have the A or B antigen on the surface of the red blood cells. Instead, they have a glycoprotein, but that glycoprotein, the O glycoprotein, isn't sufficient to evoke an antibody response. Okay, it's basically the recessive allele of these three variants of these glycoproteins that you can find on the surfaces of the erythrocytes. So an individual with type O blood is going to have both anti-A and anti-B antibodies in their plasma. So what does this mean for them in terms of receiving blood? Well, it means that they cannot receive any blood that contains either an A or a B antigen or both on the surfaces of the red blood cells. It'll attack and destroy it. So these individuals are what are called universal donors, but they can only receive blood from another O individual. Okay? The reason that they can donate blood to anybody, remember, is that the process of collecting blood involves the collecting of whole blood, but the removal of the plasma components from the whole blood before it's given as a transfusing unit to another individual. So an individual who is type O who donated their blood would end up now at the end of the day, after the whole blood is processed, with just the erythrocytes with the O antigen on the surface of the red blood cells. And this blood could be given to any of the other blood groups, O, A, B, or AB. But if an individual is type O, they're going to have in their plasma both the anti-B and anti-A antibodies. So if we attempted to give a type O individual either A, B, or AB blood, those incoming antigens would react with the antibodies in the serum of the person with type O blood, and an agglutination reaction would result, and the patient could die. Okay? So in a type O individual, you have neither A nor B antigens present on the red blood cells. You have both anti-A and anti-B antibodies, okay, shown here, and you are termed universal donor, but the only blood type you can receive is another type O individual. Okay, so that's the story on type O blood. Okay, let's look at a type A individual. Type A blood contains the A antigen on the red blood cells. They will have antibodies against type B antigen, which means that a type A individual can receive blood either from a type O individual or another type A individual. They have anti-B antibody in their plasma, they have A antigen on their red blood cells, and they can receive blood from either A or O individuals. Okay. Now, in a type B individual, what we have is the red blood cell B antigen on the surface of the red blood cells. We have the anti-A antibody in the plasma. Okay, and as a result, we can only receive blood from either a type B individual or a type O individual. If we receive blood from an A individual or an AB individual, what would happen? These anti-A antibodies would react with the incoming blood, and they would agglutinate it, and you could kill the patient. Okay? Now, an AB individual is, in a sense, the luckiest individual of all, because an AB individual has both the A and B antigens on their red blood cells, and as a result, they do not have antibodies against either A or B in their plasma. And as we already stated, there is no antibody against the O antigen. So these individuals can receive blood from any of the other blood types. They can receive A, B, AB, or O. It doesn't make any difference because they have no plasma antibodies against either the A or B antigens. Okay? So they are called universal recipients. And what you can see here is the difference in distribution of these different blood types among the different racial groups here in the United States. And what you'll note is that the most frequent blood type among Caucasians and African Americans and Asians and Native Americans is type O. Okay. Second most frequent blood type is type A. Okay. Next most frequent is type B, and the least frequent is type AB. Okay. And why is that? Well, honestly, it's luck of the draw. Okay. Essentially, the frequency of a gene within a specific population is determined either by natural selection or by random chance. And in the case of the ABO blood groups, we're not talking about any particular antigen that's going to give you a selective advantage over anybody else in terms of your survivability and the likelihood that you're going to live long enough to reproduce. So what we're looking at here with the U.S. population is something that we could kind of call founder effect, more or less. It just so happens that the majority of people on this continent happen to have the O allele, the O version of the gene, as opposed to either the A or the B version of the gene in their DNA. And as a result, now this is the most prevalent blood group. Okay? Now there's another set of antigens that we need to talk about, and these are the Rh blood groups. There are 52 named Rh agglutinogens, known as Rh factors. C, D, and E are the most common. Of those three, D is the most common. Rh positive indicates the presence of the D antigen. Okay, so somebody who has the D antigen is Rh positive. This is 85% of Americans. Anti-Rh antibodies are not spontaneously formed in individuals who are Rh minus. Okay, so those individuals lack the D antigen on their red blood cells. Anti-Rh antibodies form if Rh negative individuals receive Rh positive blood, or an Rh negative mom carries an Rh positive fetus at the point of childbirth. So you have to be exposed to the Rh antigen if you're Rh negative in order to begin to generate antibodies against this antigen. Now, this has a consequence, okay? A second exposure to the Rh positive antigen in an Rh negative individual will result in a transfusion reaction, a negative reaction. And so where this comes into play is in mismatched transfusions, or in the scenario in which an Rh negative mother has an Rh positive child. Okay, so let's talk about the transfusion scenario first. Let's say that we have an Rh negative individual. Let's just for fun say that this individual is O negative. Okay? And then one day this O negative individual needs some blood. Needs a transfusion. Okay? All right. And let's say this individual goes to the local hospital and they get a transfusion, but they happen to get a transfusion of O positive blood. All right? Well, that transfusion will work. Okay? In this case, the negative and the positive indicate the presence or the absence of the Rh antigen. So an O negative individual lacks the D antigen, while an O positive individual is going to have the D antigen. Okay? This is one of the things that confuses people when we're talking about the Rh blood groups and the ABO groups. Generally, when you have your blood typed, they check for both of those different groups. Now, the gene that codes for the D antigen is different from the gene that codes for the A, the B, or the O antigens. They're in different locations in the DNA, and they don't have anything to do with each other. Okay, they're just different sets of glycoproteins. So in an individual who is O negative, what we mean is that individual does not have the A or the B antigen. They have only the antigen that's coded by the gene little I. Okay. And that individual also does not have the D antigen. Instead, they have a gene indicated as little d. Okay, in fact, they have two copies of little I and little d. Okay, okay, so the D's are referring to this part. Okay, and the ABO system is referring to this part. Okay, now an O-positive individual is going to have a genotype of little I, okay, and they're going to have at least one copy of the capital D gene. Okay, so big D, and then in all likelihood the other allele is little d, but what we'll do is we'll just put a line here and we'll put question mark because it's hard to know what the identity of the other gene is unless we have some information from their children. Okay, so remember that now, guys, that the ABO reference is just referring now to the A, the B, or the O antigen, whereas the plus or the minus is referring to the RH group. Two separate genes, two separate pieces of information. Okay? Now, let's say this O-negative individual needs a transfusion and he gets some O-positive blood. Well, this is going to result now in an exposure of the RH antigen inside this individual. Exposed. Okay? So what's going to happen next? That individual is going to start to make anti-D antibodies. And it'll take a while for those antibody titers to ramp up. So this individual may go their whole life without any problem until, let's say, they went back to the hospital and they needed another unit of blood and they got another O-positive transfusion. Now, at that point, what would happen is that that individual would have a negative reaction. It would agglutinate this second transfusion. And the result would be massive blockage of capillaries and perhaps the death of that individual. Okay? So it's the second transfusion where we have a problem. Now, a slightly different condition exists in Rh-negative mothers who are carrying Rh-positive children. This is called hemolytic disease of the newborn, also known as erythroblastosis fetalis. So two different names for the same disease. It occurs only in Rh-negative mothers that carry an Rh-positive fetus. The Rh-negative mom is exposed to the Rh-positive blood of the fetus during the delivery of the first baby. This baby will be healthy and suffer no ill effects. However, at the point of birth, the mother's blood and the baby's blood will mix once the placenta has lost its connection with the uterus, and that fetal blood now is mixed with the mother's blood. And what happens as a result in the second pregnancy is that the mom's antibodies, which she has produced as a result of that exposure, will cross the placenta and destroy the red blood cells of the Rh-positive baby. Now, the reason that this happens is because it's a quirk of biology that the anti-Rh antibody happens to be very small. Okay, in fact, small enough to cross the placenta and cause this kind of problem. Most other antibodies are too large to cross the placenta and attack the baby's tissues. Thank goodness, because you have to remember that every child is going to be genetically different from its mother. And if we had this problem as a result of incompatibility of the antigens, what would happen now is that these antibodies could cross anytime they want and destroy the alien tissue, all right, the developing fetus. But that doesn't happen, and it's in part due to this size restriction. So the anti-Rh antibody is little, okay? It has the ability to cross that placenta and destroy the red blood cells of the Rh-positive baby. The baby is treated with pre-birth transfusions and exchange transfusions after birth in an attempt to save it. But these days what we have now is a compound called RhoGAM, which is basically a chemical that can disguise the Rh antigen. And as a result, a mother who could potentially have an Rh-positive child, if she's administered RhoGAM before, during, and after the pregnancy, we are basically hiding the Rh antigen from her immune system, and as a result, she will never generate the Rh antibody, and as a result, it will not cross the placenta and destroy the baby's red blood cells and potentially kill the child. Before this RhoGAM came about, what you used to have to do is go down to the courthouse and have a blood test. And essentially, if there was an Rh incompatibility between the husband and the wife, where the man was Rh positive and the woman was Rh negative, you had three choices. You could get married and not have children, you could get married and adopt, or you could not get married, because you ran probably a 50% or better chance of that second pregnancy failing due to hemolytic disease of the newborn. But with the advent of RhoGAM, the need for the blood test was obviated. Okay, so now anybody can marry anybody else, regardless of divergent blood types. That's no longer going to result now in a potentially disastrous pregnancy. Okay, transfusion reactions occur if mismatched blood is infused into a recipient. The donor cells are attacked by the recipient's plasma agglutinins, the antibodies that are waiting in the plasma for that alien blood. They agglutinate and clog small vessels. This causes the rupture and release of hemoglobin into the blood, which results in diminished oxygen carrying capacity, diminished blood flow into the blocked vessels, hemoglobin in the tubules of the kidneys, which results in renal failure. Symptoms include fever, chills, low blood pressure, rapid heartbeat, nausea, and vomiting. The treatment is to prevent the kidney damage using fluids and diuretics to wash out the hemoglobin in an attempt to save the individual's life. A type O individual is termed universal donor because they have neither the A or B antigen on the surface of their erythrocytes. A type AB individual is termed universal recipient because they do not have either the anti-A or anti-B antibodies in their plasma. Other agglutinogens can cause transfusion reactions. Autologous transfusion are where the patient pre-donates. Essentially what you're doing is you're giving your own blood in an attempt now to give it back to yourself in the event that you need it. Okay, so you can go to a blood donation facility and donate your own blood and basically earmark it for your own use. Not a bad idea, actually. Blood typing is generally done prior to a transfusion. It's done by mixing red blood cells with antibodies against its agglutinogens, and this causes clumping of the red blood cells if there is a positive reaction. It's done for both the ABO and RH factors. Cross-matching is where we mix the recipient's serum with the donor red blood cells prior to transfusion, and we mix the recipient's red blood cells with the donor serum. And what you're seeing here now is what happens when we have a positive or negative reaction. Okay, so here we have a type AB individual. Okay, their blood has been sampled. It's been mixed in this particular well with anti-A antibody, and look what happened. It agglutinated. Okay, that's what those little coffee ground looking specks are in the slide. It also agglutinated in the presence of anti-B antibody. And why is that? Because an AB individual has both the A and B antigen on their erythrocytes, and as a result, both antibodies will react. A type A individual is going to agglutinate with the anti-A antibody, but not with the anti-B antibody. See, there's no reaction here. A type B individual will not react with the anti-A antibody, and react with the anti-B antibody. Why is that? They have the B antigen, and as a result of exposure to the B antibody in this test, they will form now an agglutinated reaction. A type O individual does not have the A or B antigen, and as a result will not react, will not agglutinate in either of these two tests. Okay? So remember, the way these tests are done is that we take a blood sample and then we put a little bit of the blood in two different wells, okay, and then we add two different reagents, one to each well, okay. In this well we use the anti-A antibody, in this well we use the anti-B antibody, but the blood is from the same individual in both wells, okay. So hopefully that makes sense to you. All right. If we lose too much blood, we can die. Death from shock can result from low blood volume, and it must be replaced immediately with either normal saline or multiple electrolyte solution known as Ringer solution that mimics the osmotic concentration of plasma as well as its electrolyte composition. Plasma expanders such as purified human serum albumin, hetastarch, and dextran can also be used. These mimic the osmotic properties of blood serum albumin, which is a major product of the liver. This is what gives blood a lot of its water-holding properties. More expensive. This is more expensive and can cause significant complications, however. Other diagnostic tests that we can use to basically check whether or not your blood is of the proper composition include the hematocrit, which basically is a test for the number of red blood cells per volume of blood. So this tests the oxygen carrying capacity of your blood. Blood glucose tests, which check for the sugar content of your blood. So this is used as a diagnostic for diabetes. And also microscopic examination for variations in the size and shape of red blood cells, which could be indications of anemias. They could be indications of osmotic reactions. They could be indications of certain types of bacterial or viral infections. This is what we call looking at the morphology of the cell, its shape under the microscope. Differential white count is used as a diagnostic test to check for the nature of a particular infection. Remember, we talked about the granulocytes and the agranulocytes, which are components of the leukocyte population. And they each have slightly different jobs. Remember, for instance, that the eosinophils are primarily involved in allergic reactions and antiparasitic processes, while the basophils primarily respond to bacterial infection, and the lymphocytes respond to bacterial, viral infections as well as parasitic infections. So if we do a white count and we check what percentages of each of the different types of agranulocytes and granulocytes are present in the plasma, we can figure out in many cases what the nature of your infection might be. Prothrombin time and platelet counts check for hemostasis. Basically, what we're asking here is, what is your blood's ability to clot? Is it inhibited, or is it enhanced, or is it normal? SMAC is a blood chemistry profile, and it can check for liver and kidney disorders. Basically, the liver and the kidney produce a lot of plasma proteins that are vital to the normal function of blood, and if we're lacking any of these components, it could indicate a malfunction in either the liver or the kidney. A CBC checks for the formed elements. It essentially is a companion to the hematocrit and to the hemoglobin test. And this is a way for us to figure out, again, the oxygen carrying capacity of your blood. Okay, now let's ask the last question, which is, where does blood come from? Okay. Fetal blood cells form in the fetal yolk sac. The yolk sac is essentially your sack lunch, your very first sack lunch, in fact, that you ever packed. And you carried it on your journey down the oviduct to the uterus. And it was essentially filled with a protein and glycogen-rich nutrient gel that fueled your early development. but there's a sac that surrounds it called the yolk sac that contains what we call the hematopoietic stem cells. So these form in the fetal yolk sac, the liver, and the spleen as well. The red bone marrow is the primary hematopoietic area by the 7th month. Okay, so we change the site of production of blood during fetal development. Blood cells develop from mesenchymal cells called blood islands. The fetus forms hemoglobin F, which is fetal hemoglobin that has a higher affinity for oxygen than hemoglobin A, which is formed after birth. The point of hemoglobin F, as we discussed in lecture last Saturday, is that it has a greater ability to scavenge oxygen from the mother's blood than adult hemoglobin. And this, in combination with the normal concentration gradient of oxygen, where the placenta interfaces with the maternal blood supply, promotes the pickup of oxygen from the maternal blood and eventually its journey back to the fetus. Blood diseases of aging include chronic leukemias, anemias, and clotting disorders. What are leukemias? Leukemias is white blood cell cancer. Okay, basically what happens is that one or multiple hematopoietic stem cells, those that are primarily tasked with generating the leukocytes, lose their cell controls and they begin to divide in an uncontrolled fashion, and they produce white cells that aren't capable of fighting disease. Okay, there are several different types of leukemias. Anemias are basically a lack of red blood cells, and this can occur for many different reasons. We can have a problem with either red blood cell production or red blood cell destruction. In terms of red blood cell production, we might lack iron, for instance, or we might lack B12, which are important components of the heme part of hemoglobin. We might have enhanced red blood cell destruction overproduction, which would result in a decrease in the hematocrit and a loss of the oxygen carrying capacity of blood. There's also clotting disorders that can occur as we age. We can either have enhanced or decreased tendency to clot. One of the things that occurs as we age, remember, is that our heart begins to weaken and our activity level begins to drop, and so the rate of blood flow through our blood vessels begins to slow. And the risk there, of course, is the potential now for spontaneous clots, for emboli to form, and this can result eventually perhaps in the blocking of capillary beds. Remember, if this happens in the heart, it can cause a heart attack. If it happens in the brain, it could cause a stroke. These are usually precipitated by disorders of the heart, the blood vessels, or the immune system. Remember, all the body systems work together. They all depend on each other to maintain normal homeostasis, and if one system breaks down, it doesn't break down in a vacuum. It's going to have an impact on all the other body systems, and so you have to think in terms of, if something stops functioning, what are going to be the likely outcomes for the other body systems? What else could go wrong? And as we study the different body systems this term, we'll sort of touch on that when we look at different diseases that affect the circulatory system, the reproductive system, the urinary and digestive system, just to name a few. And we'll note what the impact is on the rest of the body. Okay, that brings us to an end here. Make sure and watch this lecture. Material from it will be tested on the exams, on the midterms, on the final, and so forth. And I will see everybody on Saturday.